Hi, my name is Marjory Moreau. I'm the Associate Director of the Computational Toxicology Division at ScitoVation. And today I'm going to make a small introduction on in vitro to in vivo extrapolation. The whole concept of new approach methodology was developed to reduce, refine, and replace animal use in chemical safety assessment. We all know that animal experiments are cost and time expensive. And with in vitro and in silico methods, we can be faster, less expensive, and obviously reduce the number of animal use. In vitro to in vivo extrapolation is an integral component in new approach methodologies-based risk assessment paradigms for rapidly translating large-scale in vitro toxicity assay results in the context of in vivo exposure. So basically, chemical concentrations active in experimental in vitro systems are converted to equivalent exposure levels in vivo via reverse dosimetry with pharmacokinetic modeling or physiologically based pharmacokinetic modeling. So what is IVIVE used for? We can use high-throughput IVIVE to prioritize chemical by estimating a human equivalent dose from in vitro high-throughput assays and by comparing with exposure predictions to develop marginal exposures, for example. Quantitative IVIVE can also be used in safety assessment with the derivation of risk values from in vitro point of departures. And IVIVE can be used to support the development of in vitro alternatives like dose range finding. Now, how does IVIVE work? IVIVE analysis requires an in vitro system that provides an estimate of the active concentration of a test chemical and a pharmacokinetic model to relate this active concentration to an in vivo dose that would produce an equivalent concentration in blood or in tissue. At ScitoVation, we are expert in vitro to in vivo extrapolation, and these 4 articles are an example of the kind of work we are doing. In the next few slides, I'm going to show you examples on 3 of these articles. First, NAM-based prediction of point of contact toxicity in the lung. In this project, we exposed 5 organotypic in vitro airway epithelial cell culture models with 1,3-dichloropropene or 1,3-DCP vapor at the air-liquid interface. We measured biological responses with transepithelial cell conductance and LDH release, and we used an airway dosimetry model to predict in vivo exposure levels that produce local in vitro cytotoxic concentrations. The next project is animal-free assessment of developmental toxicity, combining PBPK modeling with the repro tracker assay. In vitro screening platforms to assess teratogenic potential of compounds are emerging rapidly and show a really promising ability to predict the outcomes of in-life animal studies and to provide an attractive means for identifying teratogens and other developmental toxicants. ReproTracker is a human-induced pluripotent stem cells-based biomarker assay that is shown to reliably identify the teratogenicity potential of new pharmaceuticals and chemicals. These in vitro responses of potential toxic outcomes can be improved for hazard identification applications by introducing a quantitative framework to translate into human equivalent exposures. So the goal of this study was to evaluate whether the ReproTracker assay can predict the in vivo developmental toxicity exposure levels of drug using PBPK models describing their biokinetic behavior and IVIVE approach. Here we performed IVIVE to predict human equivalent developmental toxicity dose levels of 2 non-human teratogens, thalidomide and carbamazepine, that would correspond with in vitro concentrations associated with adverse outcomes in ReproTracker. This project is part of an SBRI phase 1 with NIHS, that we won last summer. And this pilot study demonstrated the importance of integrating PBPK models in assessing developmental toxicity in vitro, and showed that the combination of these tools can be used to improve the safety assessment of drugs and chemicals without animal testing. Now, the last project is the use of quantitative in vitro to in vivo extrapolation for the assessment of non-combustible next-generation product aerosols. In this study, we combined a PBPK model with a lung deposition model, NPPD, to better understand the pharmacokinetic of nicotine in non-combustible next-generation products and combustible cigarettes. We run the NPPD model to characterize particle deposition in the respiratory tract and develop a PBPK model for nicotine that was validated with human clinical trial data. Finally, we estimated the human equivalent concentration and predicted blood concentration based on the minimum effective concentration derived after acute exposure of B2B cells to cigarette smoke or heated tobacco product aerosol at the air-liquid interface. This publication will be released hopefully soon. It's under revision right now. So thank you for listening at me today. If you're interested to see the results of these case studies, I encourage you to look at this published article. If you are going to the SOT in March in Salt Lake City, you can also come by our booth. It's booth 606. And if you're not coming, you can just contact me and I will be happy to discuss about PBPK modeling and IVIVE. Thank you.